I would like to gratefully bring on to our virtual stage here, Steve Diver, who currently works at the University of Kentucky. Steve has done many great research projects and consulting projects. I first came across Steve's name trying to research this stuff and found he was, arguing with Elaine Ingham about facultative anaerobes back in the early 2000s. And there's still sometimes you can find these little threads just from way old soil science discussions on the internet. And Steve has been huge influence, I'd say, in modern day soil and health practices. He's extensively knowledgeable about remineralization. He's very knowledgeable about biodynamic compost and various aspects of the nature farming of Asia as well. Steve, he was helpful with touring around with Gil Carindang when there was the tour that Gil Carindang did in 2003, and helped with his teaching of workshops and stuff. And you helped publish Gil's book as well, right? Through Atro, wasn't that where it was? Or
yeah, Gil and I were co-teachers at the Eco Farm Conference in 2003.
Okay.
Which was the first, pretty much the first presentation on IMO at an organic farming conference in the country.
Yeah. That's some history right there. That's great.
Okay, so if I'm looking at, I think I've got you can kind of see the screen, and there's kind of the next slide. Is that what it looks like?
Yeah.
Okay, so I really not quite sure how to get the full screen up. So anyways, we could go this way. So let me just start here and say that it's really an honor to be with the Nature Farming Convergence. And I appreciate Peter pulling this conference together. And so what I'm going to talk about is talking about concepts and practices for soil biology and probiotics in natural farming systems. And I'm going to speak from a perspective as a consultant and a teacher, talking to a group of farmers or gardeners, and how this system is really empowering, and where it's come from, and try to tie together some principles of soil health and soil biology, and then some practical applications, that sort of thing. So I am the farm superintendent at University of Kentucky Horticulture Research Farm. And but as I am speaking from my experience working over 35 years in sustainable agriculture. So the first thing is, let's start with a meme. This is a meme off of Facebook, and it's like, what's going on? So back years ago, if you were driving down the highway in a car, your window shield was splattered with insects, and now it's not. So what is going on? And it's like Marvin Gaye. What's going on? So that points to populations in danger. So we're hearing about population crashes with insects, birds, bees. Each of these living species perform critical ecological functions in natural ecosystems. And the suspected causes include loss of habitat, agricultural pesticides, and electromagnetic frequency interference. So that's kind of a clue of what's going on with agriculture and its impacts. Agricultural impacts include habitat destruction, soil erosion, desertification. This is going on a worldwide basis. It's really disrupting the ecology of the planet. You have kind of a classical aspect of modern industrial agriculture is monocultures. And so with monocultures, and it's across the board, when you have that, you do lack diversity and ecosystem resilience. So what that points to is that whereas for sure, modern industrial agriculture is very productive, and it's really solved a lot of problems with hunger around the world, et cetera, it does have some unintended consequences. And basically, we are externalizing the costs of, this kind of widespread agriculture. And that includes the runoff of nitrogen and phosphorus into waterways that go into the Gulf of Mexico, other parts around the world, and creates dead zones, lack of hypoxia. The widespread use of pesticides lead to endocrine disruption in both male and females. There's a saying now that for young men, you are half the man that your grandfather was because of decreasing sperm counts around the world. So another angle is the GMO technology package with GMO seed and over-the-top glyphosate herbicide that controls weeds. Unintentionally disturbs the monarch butterfly populations, and it disturbs soil biology. So what that's going to lead to is that is that all we get? I mean, is that the only option, or is there something else? And that leads to this slide on alternative farming systems that I like to share with farmers to point to people that it's not just about organic versus conventional. There's been a number of alternative farming systems that have come up over many decades and pretty much all over the world. And some wherever you are, there's farmers trying a different approach. And so that includes things like sustainable, which is really big with USDA, the Sustainable Agriculture Research and Education Program, with the USDA NRCS program and their soil health program. And that pretty much got started in the early 1990s and really took off. It's a real umbrella style of a system. Is not a one kind of farming practice. It just encompasses these principles and practices. Organic includes both the larger kind of organic agriculture as well as certified organic, which is more codified under the USDA National Organic Program. And then permaculture, which is a worldwide movement based on ecological design, totally mimicking nature and creating patterns in food production and built landscapes and edible landscapes. And then you have these nature farming movements from Japan and Korea and India that are very influential. We're going to revisit those here in just a little bit, but each one of these have various on-farm biomass resources that have been transformed and fermented into a number of different substrates that can function for soil fertility and plant health and for pest control. And then it's really big in the United States is holistic grazing integrated with crops. And really, if you think about every agricultural civilization that has sustained itself over thousands of years, they've always integrated crop and livestock. And then in Europe, in United States, Australia, and in other parts of the world, biodynamic agriculture has been really big. And it's well known to use the biodynamic preparations. And then another really big one is eco-agriculture. There's more acres managed under eco-agriculture in the United States than there is organic farming. Very influential with Acres USA and some pioneers. But what's important to note is that each one of these alternative farming systems have common themes. And one of them that is that peat farmers are aiming for local healthy foods and food production systems. They're striving to attain non-toxic and least toxic pest control. And a lot of that is because these farmers are designing agroecosystems that mimic nature, and they're taking advantage of these ecosystem processes. So a big emphasis on biodiversity, on mulches, on less soil disturbance, intercropping, cover crops, and many other aspects. And then a big reliance and emphasis on organic and biological soil amendments. And all of that, those last items, then they lead to a healthy consortia of beneficial microorganisms. So what we could say here is that this is really helpful to put this into perspective with a bell-shaped curve, and where this is kind of the classic model where you have innovators and then early adopters, and then you have the early majority, the late majority, and the laggards. And so this model actually was developed to describe agricultural practices and adoption. It's been around for decades, but it's a real good illustration of what we're talking about. So yes, a lot of these alternative farming systems, they've been envisioned by pioneers, they've been spread by teachers and consultants, and then there's a broad swath of farmer practitioners who have adopted these. And so they're definitely on the innovators and early adopters scale. So if we look back, there's some really significant reports, like the Report and Recommendations on Organic Farming, published by USDA in 1980, and the Alternative Methods of Agriculture, which came out of Europe. Both of those were roughly 40 years ago. Then the first International Conference on QSA Nature Farming and EM Technology in Thailand from 1989, roughly 30 years ago. And then Han Kyu Cho's first book, very rare, obscure book on Korean natural farming and indigenous microorganisms in 1997, roughly 25 years ago. So that's a good indicator of that. So that's kind of what we're talking about. And so that's available. It's still cutting edge, even though they've been around that long. And we're still learning. We're still passing on that information and adopting it. So it's a slow process of making changes and spreading this around, but it's getting better. So let's look at a few examples. So let's review QSA Nature Farming and EM Probiotics. This was the system envisioned, developed by a horticultural scientist, Dr. Teruo Higa in Japan. And that is a microbial inoculant, EM effective microorganisms. And it's syntropic, it's regenerative, it's very multidimensional. It is not just a crop biostimulant, which is what a lot of people think of when they think of a microbial inoculant. This is very versatile. So it's, some of these different things, for example, you got the EM inoculant. There's also EM Bokashi. You can make activated EM. You can make fermented plant extract, fermented fish. You can make an herbal vodka pest control. You can use it for antioxidant human tonic and to improve your gut flora. And then there's also antioxidant ceramic powder. And there's been a number of different key publications that have come out on that. But for example, Earth Saving Revolution that Peter mentioned, then a whole series of QSA Nature Farming conferences, and then Life in the Soil video. Those are all been really well known. The next one I'm going to mention is Korean natural farming, which came out of Han Kyu Cho's work in Korea that Peter mentioned, and very unique. A lot of similarities in some ways to the Japanese QSA Nature Farming in terms of using microbial inoculants and on-farm biomass resource to make these extracts. So, but in this instance, instead of starting with the mother culture that's manufactured, you use on-farm indigenous microorganisms. So that's what this indicates up here. You have IMO, 1, 2, 3, 4, 5, fermented plant juice, fermented fruit juice, fish amino acid, lactic acid, bacteria serum, oriental herbal nutrients, brown rice vinegar, and so on. And then there's a number of books there. This has really taken off in certain parts of the country. Hawaii is really big. University of Hawaii has adopted this. There's been some instrumental consultants and scientists over there. There's two gentlemen who have really become really great teachers and are sharing information on this. Eric Weinert in Hawaii, and then Chris Trump in Idaho. They've taught a lot of workshops. There's a lot of YouTube videos on that. So a lot of farmers and gardeners are adopting this. Next one is Jadam, low-cost organic farming. This is Yongsung Cho, who is Master Cho's son. And so he's developed a kind of a spin-off system. And he phrases that as low-cost organic farming. And there, again, a lot of different recipes developed from on-farm biomass resources. Indigenous Microorganism Solution, JMS, and then JWA, Jadam Wetting Agent, and there's the sulfur and herbal solution and pesticides and so forth. And he's published a number of books and videos. This one's a really dynamic and powerful system to add to the first two. And then natural farming in India, some of the pioneers there include Subhash Palekar, who's going to be one of the speakers, but also Bhaskar Save. And I was actually at Bhaskar Save's farm in India and had a conversation that it took, I think it was translated 3 different ways, because I could speak English and my friend could speak Tamil, and then they could speak Hindi or something like that, and then that person could speak to Bhaskar Save, who was in Gujarati. So, anyways, that was great. And then, but natural farming in India is particularly interesting because it is based on Vedic agricultural knowledge that is at least 1,000 years old. It again relies on on-farm resources like ghee and honey and leaves and cow dung and cow urine and cow milk and so forth to make a number of these different recipes. And so those are all being practiced by farmers in India. There's some books down there that speak to some of this. And big impact. Farms in India are really interesting. They've got, compared to the United States, the farm on India has really good quality cow manure and cow products to work with versus over here where they're largely contaminated by fly insecticides and worming agents and so forth. So that's a good one to know about. And then the other one there is biodynamic preparations. That is the horn manure, the horn silica, BD 500, BD 501, and then a few others like the barrel compost, also known as cow patty pit, and then a whole bunch of liquid herbal manures that are made from comfrey and yarrow and different on-farm herbs. And using different manures, silica, which is quartz, and herbs, animal parts, and local plant and herb materials, you can do these different fermentations on the farm that have these biological and energetic attributes. But indigenous microorganisms definitely play a really big role in transforming these products into useful substances that drive farm fertility and food quality and health and so forth. So, and then, you know, you see a big emphasis there from the Asian natural farming systems, but on a worldwide basis, agroecology, agroecosystems, integrated farming systems, no-till, intercrop, cover crops, agroforestry, allocropping, syntropic agroforestry, all of these, like I said, are designing agroecosystems that mimic nature to take advantage of these natural process and interactions that you can do on the farm. So when we talk about natural ecosystems, let's talk about a couple of them. So you have forests, you have prairies, you have wetlands. Those are the big 3 natural plant ecosystems. And if you look at the forest, I mean, it's got a variety of trees. The soil is always covered with an incredible diversity of leaves and wood and logs, with just teeming with life. And so the forest trees have moisture, they have mycorrhizal fungi, and nobody is fertilizing the forest. And yet look how vast and productive the vast boreal forests are and tropical forests are around the world. In the same way, the prairie is an amazing ecosystem with hundreds of different species of grasses and legumes and forbs that we call wildflowers. And a prairie can withstand drought, it can withstand flood, it can have seasonal moisture. And there's some kind of species that will survive and thrive in that. And they have incredibly deep roots. They're long-lived. Lots of cellulose and plant matter is deposited on the soil. Incredible diversity above ground and below ground soil food web. So these ecosystems, again, are driven by these natural ecosystem processes, which, for example, is the soil food web interacting with the soil, interacting with clay humus and the rock mineral substrates to make minerals available to plants, and incredible biomass production and health and beauty from these natural systems. That is the inspiration that natural farmers draw from and learn from. So I'm going to go through some slides on soil health and soil biology for, again, like I said, this would be for farmers and gardeners wanting to know about this, and I'm going to point out some key points here. And so let's look at characteristics of a healthy soil. So you'll see, for example, good soil tilth, it's friable, it's easy to work, it's got good physical condition with soil organic matter. It has a lot of beneficial microorganisms, both macro and microorganisms. It has stable soil structure. And then what's interesting is last one down here is that these model agroecosystems can result in remarkable paradigm shifting regenerative agriculture. Farmers are just blown away at how well they can grow crops by mimicking nature, adopting no-till, adopting heavy mulches and cover crops, multi-species cover crops, and intercropping and so forth. And so, and one of the other aspects of that is that Dr. Terrio Higa came up with this term, which is limit breakthrough. And what he's saying is that with the adoption of these unique soil biological systems and microbial inoculants, they're seeing this in QC nature farming. And Master Cho is describing the same thing in Korean natural farming, where they're seeing things they've never seen before. There's more grains per rice head. There's more fruit on a banana stalk. There's more fruit on a pepper plant. And it's all driven by these biological processes. So now let's go to a few slides that I find very interesting that I put together. And that is, let's start with, what does all life on Earth, humans, zebras, lichens, slime molds, jellyfish, flowers, bees, and soil microbes all have in common? And what they have in common are cells. Cells are common to all of these life forms. And what's interesting when we talk about cells is the conversion of sunlight into carbon-based molecules by both plants and microorganisms, and the ability to manufacture millions of life forms, including bacteria, algae, protists, fungi, plants, and animals. And in plant cytoplasm, there's the ability to produce over 200,000 carbon-based metabolites. These processes take place at warp speeds. They take at nano, pico, and femtoseconds, which are 1 million to 1 quadrillionth of a second. And they take place in the micro nanosphere, which is 1 millionth to 1 billionth of a meter. And then when we look at, let's say, a bacterial cell or a yeast cell, some facts that I find interesting is that it's helpful to think about cells as little factories that have administration, they have security, they have procurement, manufacturing, transportation, waste management. Inside one bacterial cell, you can have over 5 million DNA, 3 million proteins, 50 million lipids, just astounding. And then yeast, you can have 120 million DNA, 100,000 ribosomes, 100 million proteins, and 1 billion lipids. And so every single molecule in nature, every plant and animal will eventually be decomposed by unicellular and multicellular microorganisms. So this slide is helpful to put in perspective what we're talking about when we talk about cell size. So let's start with a meter, just a little more than 3 feet, which is kind of the size, for example, of a child. And then about 1,000 times smaller, that is a millimeter. And then 1,000 times smaller than that is a micrometer. So now you're looking at 1 millionth of a meter. And so you can see where the cellular organisms exist. and you're down to the micrometer size. You're now inside the cell, and then 1,000 times smaller than that is a nanometer, which is 1/1000th of a meter. And that is subcellular substances like we talked about, like the DNA and the lipids and the proteins and so forth. So that helps to put in perspective. The next thing is that let's look at the composition of a bacterial cell, and talk about cellular conversion of CO2 or carbon to various metabolites and compounds. So let's see, you've got a cell is composed of 15% protein, you've got small molecules, you have RNA, you have phospholipids, DNA, and polysaccharides or sugars. That shows you the composition of the cell. And then, so it's making a lot of these products to sustain life and grow new cells and decompose and create new things. So some of these that are interesting are important to understand in agriculture and soil health are amino acids, enzymes, organic acids, hormones that stimulate plant growth, polysaccharides or sugars and sticky things, and vitamins. There can be over 1,000 different enzymes in a bacterial cell. So another thing about the soil microbial world to put in perspective is that everything once living, every plant and animal that was once living will be consumed by microbial decomposers. And every kind of microbe occurs everywhere plants and animals exist. And the other thing is that every surface, every living and non-living surface on the planet is a microbial habitat. So we talked about enzymes. Enzymes are proteins that are produced by microbes and other cells that act as a catalyst. And so you have a substrate, you have a compound such as some biomass that are generated by plants and animals that are acted upon by enzymes. And you have soil enzymes, you have intracellular, extracellular enzymes, and so forth. But if you want to convert and transform starch, glucose, cellulose, chitin, phosphorus, proteins, lipids, there is an enzyme that is responsible for helping to convert those. And so those are really important characteristics that are generated by microbial cells, and farmers take advantage of that in natural farming. So the other thing are organic acids. Organic acids are compounds that possess acidic properties. They have key functions like solubilization, mineralization, biological control, detoxification, chelation. And so you have lactic acid, acetic acid, citric acid, oxalic acid, malic acid, and so forth. So if you're going to have rock dust, those are relatively inert materials. If you have rock phosphate, those are relatively inert. But if you put those in a compost pile and you have incredible amount of microbial activity, they're generating enzymes and organic acids, they're solubilizing and mineralizing and chelating those materials into organic complex materials like compost humus that then become available for plant uptake and plant use. So let's move on and talk about soil biota. And this is kind of how, this is a really great way to classify them. This is pretty classic, is that you have 4 broad groups. You have microflora, which are very small, and that include bacteria, fungi, actinomycetes, and algae. So that's what we're talking about when we talk about microflora. And then microfauna, which these are all microscopic. These are little animals, and they're larger. That would include protozoas and nematodes, or the protists. And then you have animals that are a little larger than that. Some of them are microscopic to you can see with a hand lens or the naked eye, and that's mesofauna. That includes mites and springtails. And then macrofauna, which you can easily see, millipedes and earthworms. But what's important is that you have microfloral decomposers, microfaunal grazers and predators, you have macro shredders, you have ecosystem engineers. And that's a quick synopsis of what we're talking about when we talk about soil biology. The other thing is the size of these different microorganisms in relation to the soil. So when we say soil, the primary soil particles are sand, silt, and clay. And so you can see the relative size of these. And sand, again, you can see with the naked eye. Silt gets very small, and clay is extremely small. So we're talking about 1/1,000,000th of a meter there with clay. But if you look at bacteria and fungal hyphae, they're in the same sphere of these primary materials. And then you have plant hairs, plant roots, and then you have soil aggregate materials. And so that's really helpful to put in perspective, because that is what's happening in the nano world. And then this is just to show you what we're talking about. This is bacteria. These are the Actinomycetes, which are really bacteria with threads that look like fungi. You have soil algae, you have fungi. And so, yeah, we're talking about tens of thousands of species of bacteria. We're talking about millions of species of fungi. So each one of these species has a role to play. They do something unique. They do multiple functions. This is some of the protozoas, the nematode and the protozoas, and then you have the mites and springtails, and then you have your centipedes, worms, and so forth. So that helps to see what we're talking about. Now, what's a really great principle to understand is that soil microorganisms live in association with plant roots and excrete sticky substances. So we talk about symbiotic mycorrhizal fungi, and we have endomycorrhizae that germinate in the soil when a plant roots comes by. It grows into the internal part of the plant, and then it sends these fungal hyphae out into the soil to forage for nutrients and bring them back to the plant. And the plant provides carbon to the fungi, and the mycorrhizae provides many benefits to the plant. So in the process of that, they excrete sticky substances, including glomalin, which is a glue-like glycoprotein. And then you have bacterial rhizospheres that excrete extracellular polymeric substances, or EPS. And then ectomycorrhizae, this is an example of what grows on conifer roots like pines. They grow on pecan, on oak, on birch, and so forth. And this is more extracellular polysaccharides. So these sticky substances are really important. Let's take a look, for example. These are some primary soil particles. This is like the sand, silt, and clay. Here's some clay, very, very small. And what you can see are Actinomycetes filaments that are actually holding together and binding clay particles together. Here's another example of that. And then here's fungal hyphae enmeshing a soil aggregate. And here's a bacterial colony on humus aggregate. So this happens at a micro nano scale, like we talked about, but these sticky substances are critical for holding the soil together and starting a chain of reaction that builds a healthy, fertile soil. So this is when we talk about soil micro and macro aggregates that we're talking about. You have a small micro aggregate, it's bound into a macro aggregate, and then a lot of these go into a soil clod, and you have plant roots in there, you have fungal hyphae, you have all this debris, all these sticky substances holding it together. And that shows you what the binding forces are. You have a lot of bacterial glues, fungal hyphae, mycorrhizal glomalin, polysaccharides, plant hairs, and so forth. So when you put all that together, you end up with what this is known as. This is what a good looking soil is, good soil structure looks like. What farmers say is black cottage cheese. And that's a really good, healthy looking soil right there. That's bound together. And what's another thing that's helpful is to go back and look at some of the old USDA literature, because it explains it so well. So from 1952, they have this agriculture information leaflet that talks about crumbly or cloddy soils. And what they did was they went out to a cornfield and they took soil from the cornfield. And this is back when corn was probably row cultivated on 36-inch rows. So you have a lot of exposed soil right there. And then they went to the fence line. The fence line is where they had some virgin prairie soil. And so they took the soil, they put them there, and then they put them with water. And what they found was that the virgin prairie soil, when it was mixed with water, held together. This is called water stable aggregates. That's what you want. And then the cultivated soil, because it's lost its integrity, because it's been cultivated and they didn't do enough organic matter management, it dispersed and turned into a puddle. And so that's what we're talking about when we talk about soil health. And so this is a consequence. Here's a field up here that you've had a rainfall event, and instead of good infiltration, you have standing water. And with just a little bit of slope and standing water, instead of water infiltrating into the soil, it leads to erosion. You have soil erosion, and you're losing soil off the farm. You're also carrying nutrients that are going into waterways. So this is a modern demonstration that's really popular in soil health and with NRCS. And so in this instance, they've taken a tilled cover crop, they've put it into a water infiltration study. They added water to it, and water ran down and created a lot of runoff that's dirty water. They had a tarp cover crop. They still had some runoff, but the water's clear. And then they had a field that was last tilled 22 months ago, and it has a mulch crop. Very little runoff. It's actually infiltrated into the bottom pan right here. And then here's the border of the hedgerow where it's not been tilled. It's like the virgin prairie, but it's like a hedgerow. And again, no runoff. It's all infiltrated completely down into the bottom pan. So that's really key to understand why we talk about soil biology and soil organic matter. So now I want to run through what I call 6 foundational principles for agroecosystems. Let me just jump in here. So one principle is that the principle of microbial abundance and diversity, also known as microbial abundance and diversity, and that is that this principle says that the population and abundance and complex diversity of soil microorganisms drives soil functions, microbial density and diversity. So that can improve soil structure, soil organic matter, soil humus, it can therefore retain soil moisture and promote soil fertility and disease suppression. The next one is the principle of soil organic matter functioning. And we talk about the 3 components of the soil, the physical, chemical, and biological components. But what's important is that soil organic matter links all 3 together. And there is a principle from biodynamic agriculture called the Humus Law that Alex Podolinsky in Australia came up with. And that the Humus Law says that the soil organic matter should be at least 3% or 5% organic matter in the soil for the natural processes to happen and work on the farmer's behalf. So the next one is soil food web or soil microbial community functioning. So this one recognizes that you have the root sphere or the rhizosphere and the leaf sphere or the phyllosphere, and these are the zones of activity and influence of microbiology. They also have plant growth promoting rhizobacteria. You have symbiotic and saprophytic fungi. All of these lead to ecosystem processes or ecosystem services. So for example, you have bacterial and fungal decomposers, nitrogen fixation, carbon fixation, transformation and availability of nitrogen, phosphorus, and sulfur, phosphorus solubilizing bacteria. You have suppression of disease in insects, production of phytohormones and bioactive substances, organic acids, soil enzymes, and all these polysaccharides like slimes, glues, and cements. So these ecosystem services are a huge benefit of managing for soil biology and soil organic matter. And then here's an example. This one is key. This is an interesting thing. This is called a pathogen inhibition assay, bioassay. And this is a Petri dish with agar on it. The middle cotton ball has the pathogen, Phytophthora. The other cotton balls have water. And because the pathogen has no competition, it grows freely across the Petri dish. In this other Petri dish on the right, the center cotton ball has the pathogen, Phytophthora. The outer cotton balls have been soaked in a worm compost extract, which have a rich diversity of microbiology. And because they generate their own sphere of biological influence, there is no competition. There is no invasion of the Phytophthora. They're able to fight off. So there's different terms for these biological control mechanisms, but this one in particular is known as competitive exclusion. This concept of competitive exclusion is key to understand some of the functions of these probiotics like EM, effective microorganisms. And then the next one I want to share is the MIND concept, microbially enhanced nutrient delivery. And it builds on the other one we just heard of. And that is microbial density and diversity in the leaf zone and the root zone influences the availability and uptake of and efficiency of nutrients. And so what farmers are finding out is that by blending microbes and minerals in things like fertigation and foliar applications, they're making these nutrients more bioavailable, and they're able to reduce fertilizer inputs from 25 to 75%, even sometimes 100% elimination of fertilizers. So the MIN concept is really a key one to remember. And then I think the last one is on root exudates and how plants are able to photosynthesize. And then, somewhere between 30% to 50% or more of these photosynthetes are leaked out into the root zone to feed the soil microbiology, which in turn has benefits for the plant. So for example, you can have amino acids, organic acids, sugars, vitamins, nucleic acids, enzymes, and ions and gases all leaking out of the roots to stimulate the soil microbiology. And so that leads to the 4 R's: Rhizosphere, Rhizo Deposition. Rhizo Deposition is a description of root exudates. The rhizo sheath is the soil that clings to the rhizosphere from these polysaccharides. And then rhizophagy is a rather newer phenomenon in plant science, which recognizes that plants absorb microbes into the roots. And then the roots can exit back out into the soil. And then they can come back in, and then a certain number of these microbes are turned into pure protoplasm and are actually absorbed by the roots as a form of feeding on microbes for nutrition, which is pretty wild. So that little segue or segment kind of describes the ecosystem processes that are important to know. So now I love this little illustration. This is some of the farmer-friendly illustrations that have come out of nature farming. This one is from Dr. Higa on the functions of effective microorganisms in the soil. And if you look up here, you see organic matter, you see the plants, you have decomposition by bacteria, you have a whole lot of things that are really going on. You have mycorrhizae, nitrogen fixing bacteria free living. You have photosynthetic microorganisms like phototrophic bacteria and blue-green algae. You have antibiotic producing bacteria such as Actinomycetes. You have other organisms like yeast that produce bioactive substances and dissolve organic matter, lactic acid producing bacteria, and so forth. And then see, they're fighting the little pathogenic bacteria. So all of that is really helpful to understand. There's a lot going on. And I will say, if we just look back, for example, of this 50-year time frame, agriculture has totally transformed from a huge reliance on chemical inputs to more and more farmers adopting these practices, these agroecosystem processes, to take advantage of, the function of bacteria and microbiology to promote plant health, to promote fertility and natural insect and disease suppression. So that leads us to the last section. We're going to run through some slides on probiotics. So for example, EM, let's talk about EM, effective microorganisms. Let's talk about indigenous microorganisms. And so we're talking about probiotics for human, animal, and environmental health. There's a lot to be said about fermentation technologies. It's helpful to understand differences between aerobic and anaerobic systems. And then understand there's multiple uses of these in treating soils and crops, livestock, organic waste. It's not just one thing. And the ability to transform raw biomass resources into biofertilizers. Let's see, this one's not happy. Oh, there it is. It's got to come, it's got to load up. Okay, I kind of forgot that. So let's start off with what you might find in a health food store. People hear a lot about probiotics. And what that implies is that these are organisms or substances which contribute to intestinal microbial balance and health, basically beneficial and effective microorganisms. So Lactobacillus, kefir, Saccharomyces, Streptococcus, all of these are really beneficial microorganisms that are helpful in gut flora and human health. And then, let's see, I got to load this one. This one, another thing that you may find in a health food store, which are now very popular, are kombucha and kefir. And so these are known as scobies, which is a symbiotic culture of bacteria and yeast. And these are really funny little substances, but that's what is driving the production of these probiotic drinks, is these cultures of bacteria and yeast that feed on some substance like sugars and teas, and transform those into beneficial probiotic drinks that have antioxidant properties and other, health properties. So when we talk about biofertilizers, we're talking about taking, again, these raw organic biomass sources that on the farm. So that can be plant materials, herbal materials, fish guts, some kind of manures, and by using microbes, either purchased microbes like a mother culture in EM or local indigenous microorganisms like IMOs, mixing them with food, sugars, and going through a fermentation and extraction process, we can arrive at a whole number of bioavailable nutrients, chelated nutrients, bioactive substances, and a whole bunch of beneficial microorganisms. So that's the key concept here. And so, for example, here is a fermented plant juice that is made from some herbs at one of the sustainable centers in Oklahoma. And that was some herbs grown on the farm that were mixed with sugars and turned into a fermented plant juice. And again, it's got all these different substances. And we looked at it under the microscope, it was loaded, totally loaded with bacteria, beneficial bacteria. Then it's important, I think, because of the history of compost teas and the reaction to anaerobic systems, that it's been confused. So I want to clear that up, and that is, it's really helpful to understand the difference between two kinds of different anaerobic processes. One is putrefactive. That's the one we don't want. And that's very common. You have a pile of fish guts, you have a pile of food waste, you have raw manure that's stacked up and left to sit outside in the hot sun with no management. That leads to putrefactive odors and harmful substances. You can smell them easily: hydrogen sulfide, mercaptans, ammonias, and they lead to the smells. They attract flies and vermin, and they can generate, as a result, they can result in pathogenic organisms. But the same raw materials, these fish guts and these highly putrescent food waste and raw manures, if you manage these, you can, with the system, with the microbes and the sugars, and you can create a fermentative anaerobic system and transform these materials and produce useful substances like the enzymes and amino acids, the organic acids, antioxidative substances, and they result in beneficial microorganisms. And so the other thing that's helpful is to talk about 3 different microbial pathways in organic matter decomposition. So the first one is aerobic thermophilic. That would be like a compost pile where you have raw manure, say some food waste, but it's mixed with straw and some leaves in the proper proportion. It's built up and it generates heat through biothermic heat, and it goes through a breakdown and buildup phase and turns into humus compost. The other one is, we just talked about putrefactive anaerobic, where it's stinky and it's not managed. But then is, for example, if you turn around and manage that and turn that into a Bokashi system and ferment it, then you're generating a beneficial product. So that's really key: putrefactive anaerobic and fermentative anaerobic. So we got just a quick review of a few things, and we talked about EM. This has been central in nature farming, but it's spread across the world and it's just been widely adopted. So farmers use it, in context of nature farming, but also just because it's a good natural probiotic. So it's classically a consortia of phototrophic bacteria, lactic acid bacteria, and yeast, but Actinomycetes are very common in EM. And then Bokashi is really a popular use of EM. This is EM brand Bokashi, and you have your kitchen food scrap bucket. You're chopping in the kitchen, you've got your peelings, and you put them in the bucket, cover with some brand Bokashi, and it goes through this fermentative anaerobic process and keeps that material in a fresh condition instead of turning putrefactive, and then that can go out into the soil. It can go out into the compost pile. So another new usage, which is really unique, it's come around the last, I don't know, 5 or 6 years, is the probiotic farmers have started taking Bokashi, brand Bokashi, adding agricultural amendments to the Bokashi process and using that to promote plant growth. And so in this instance, this is at our research farm. This is some research I did. And that is, it was an ag brand Bokashi with lactic acid bacteria, wheat bran, biochar, azomite, which is rock dust, humic acid and kelp. And then it was put into a peat-light mix, just a standard pro mix, BX, at 1%, 3%, and 5% with this Bokashi material called Biokashi. And this is the growth curve. So that was a really awesome demonstration of the power of a little bit of microbial additive to a potting mix substrate. And so, like I said, we got really good growth response in that research. And then finally, this is some work that Nicola Maddock has done up in Ohio. He and his brother have a 40-acre hops farm. This is an example of some outdoor indigenous microorganisms, or IMO, some indoor IMO, some beautiful substances they've cultured from the forest floor, duff, the soil from the forest, the leaf litter from the forest, that have been fed white rice, and then the native microflora have grown onto that. And that leads into the next recipes, and they've done large-scale IMO3 with barley, rice bran, and wood chips, and then they've made IMO5 they've made it with IMO4 and horse and chicken manure. They've got a compost turner, and that is the basis of the soil fertility to manage that 40-acre hops farm. So. And then a few slides, Jadam, a bunch of different things going on with Jadam. Here's a couple cool ones. That's a cucumber leaf Jadam liquid fertilizer This is a calcium chitosam Jadam liquid fertilizer with crab shells and shellfish. So you know you're going to get some good benefits out of that for fungal control with the chitosam, the chitin, and you're going to get good activity there. Then, yeah, I'm just going to run through. This is another, again, it's these probiotic systems are again, have become widespread. This one has been popular down in South America. This is a biofertilizer that's done in a barrel with manure and yeast. And then here's the instructions on how to do it. You can see a lot of this online. These guys have taught a lot of workshops all over. And then they use that for soil and foliar applications to stimulate plants. And then it's really taken off. And again, this is a good thing that these technologies have grown beyond strictly nature farming and have become more widely adopted by farmers because they work. So these are some microbial fermentation tanks down at Latin America farm tour. And this is an operation where they're doing a ton of different fermentations and extracts and developing products for both fertility and pest control that farmers are using. So yeah, that brings us to the close. And yeah, hope that was helpful. All right, so let's see, Peter, are you around?
Yes. Yes, I'm right here.
Good. So it actually worked, huh?
Yeah. Yeah, it was great. Thank you so much. It was super awesome. And just to have the perspective of someone, like you said, since 1995, you've been knowledgeable or learning continuously about EM microbes and other microbes. It's awesome. It's awesome to hear your perspective from just a longevity standpoint of how this has changed and how we have moved forward and adaptations and innovations and cross-pollinations of the fermentations.
Great. Well, it's so exciting, and it's hard. It just 1 hour presentation was. I'm just trying to. I even have some of my garden clients I think are on the conference call, and people will see this on YouTube. So again, this is a perspective I'm just sharing from a farm consultant. I'm trying to honor the work that many people have done all over the world, and I'm definitely still a student myself, and but I will say that I've also done a fair amount of this on a pretty good size scale. So it can be applicable at the small scale as really at the broad scale level. It's really very empowering. It's totally transformed our concept of sustainable agriculture and the ability to get out there and do things. So it's just right for more people to try out. The universities are great because they can bring scientific knowledge and science to bear, but the farmers are really leading the charge on this.
Right.
And that's for sure. The practitioners are the early adopters and innovators.
So I have a question, because I've had some very interesting experience getting these microbes on my skin. I'm not much one for drinking. I've never done a Bokashi bath or anything, but have you ever had any like psychobiotic, like psychedelic almost experiences from handling large brews of these microbes?
Psychedelic, I would say some real health benefits for sure. Yeah, and just as an example, EM is well known for being brewed or fermented with the intention of a human probiotic drink.
Right.
And one of my
pasteurized version is sold. EMX is pasteurized, so it's they just promote the antioxidant benefits. But I did hear actually even that because of some of the claims that Higo is making in an Earth Saving Revolution 2 about the antioxidant miracles basically happening from what he saw with EM, that he was censored by the government because of maybe he was leaning too far into alternative health information. Their version of the FDA kind of might have shut him down or something. But anyway, I've had some experiences where I'm just like doing an IMO culture, or the first time someone gave me a bottle of homemade EM1, when I got it on my skin, I felt that inoculation, and I feel I just more like coherent as a thinker from the microbes kind of dialing in my own gut brain access. So that's one, like, on the human health benefits, like that's where I really, the psychobiotic, gut brain access side of things is where I've just noticed, like, that's where it ties into how we are cultivating and perfecting the human being, like Fukuoka said, trying to move from dysbiosis into eubiosis and mental health.
Yeah, sure. Since you brought that up, let me just mention, let's go ahead and talk about EM fermentations for human consumption. And so the couple people to mention there, the EMX for sure, the companies that are the master brewers of EM, they do make the EMX fermentations, but on a kind of just do-it-yourself scale. There's two people I should mention. One is one of the guys that is a friend of mine, and we did the workshop in Shepherdstown, West Virginia, back a long time ago, is Vinnie Pinto.
Yeah.
Vinnie Pinto's book was real instrumental in promoting and expanding the concept of EM recipes. And I'm talking about super EM recipes, where the recipe includes some additional substrates, various sea minerals or rock dust, various fruit juices and herbs, that because these different microbes will feed on these other substances, they'll create additional antioxidants, and they you can make them food grade and you can drink them. So the other one is, I do want to mention another effort in this area is the company called Vitabiosa.
Right.
And Vitabiosa was invented by a Danish farmer who actually lives in Canada now. But you think about Danish, you think about Ukrainian farmers. These are the kind of folks that would use different berries and herbal things for health. And so Vitabiosa has a bunch of different herbs in there that are fermented with EM mother culture and make a fantastic human probiotic drink. Yeah. And so let's talk about antioxidants. Again, my presentation was fast. But a key concept that we should mention is antioxidants. So antioxidants matter in a lot of different ways, but just the most simplest example would be to take a nail and put the nail on a moist paper towel with plain water and let it sit for a couple of weeks. Take the other nail, put it on a moist paper towel that has been moistened with EM inoculant, and let it sit for a couple of weeks. And you can put them in, like, for example, a plastic tub or something, or container. What you're going to find is that the nail with plain water is going to start rusting. The nail that has been treated with the EM microbes will not rust because it has antioxidant properties. And when you say antioxidant, you're talking about stealing electrons. And right. In the human physiology, that creates free radicals, which lead to degenerative diseases and health issues. So the opposite is the antioxidative are giving electrons. They're powerful reducing agents. And so that leads to benefits for human health. But in probiotic treatments of environments, this is why these organisms are really helpful in controlling odors and improving animal health in barns and in CAFO operations. You can take a CAFO operation, like a confined animal feeding operation of poultry, of hogs. You can totally transform and enhance the health of those animal environments by using these probiotics in the drinking water, in the animal feed, mixing them over the bedding. And because they have these powerful phototrophic microorganisms that have reducing power, they are antioxidative. The same way with the lagoon, with the dairy lagoon and hog lagoon. These things are really huge and very stinky. You can totally transform these lagoons into a healthy, pool or lake of manure, liquid manure. because of these properties. Yeah. So I do want to share that.
So it almost like this has been what I've been working on in the last year is basically reducing it down to free radical farming versus antioxidant farming. Like nature farming would nestle definitely in the antioxidant farming category, and so would a lot of other regenerative ags, and then a lot of the conventional chemical-based heavy tillage ag, that's all kind of free radical farming, like kind of tying into John Kemp's plant pyramid nutrition thing, with fats and oils being the highest form of photosynthetic energy storage. And then, like you said yesterday about Maria or Anna Maria Prima Vici, saying, you know, the infrared spots on the leaves is then where the plant senses like the weak alcohol product, or the insects see the weak alcohol production on plant leaves. And
yeah, so I'm looking through some of the questions and things like that.
Okay.
So Lala Gardens, which is I see on Facebook, doing some really great things with Korean natural farming and probiotics. They want to know about the book that keeps coming up. So that's the Earth Saving Revolution book by Higa. So if you look on Google Images or Amazon, it's the Earth Saving Revolution. There's actually 2 of those. Yeah, that's a good one. That's a good one. Let's an example from that, from the, you know, we'll try to talk a little about everything, but let's go ahead and talk about the Earth Saving Revolution and Higa's experience. And so we talked about these dead zones.
Right.
And these dead zones occur not just in the Gulf of Mexico, but around the world, where industrial, you know, cities and agricultural dump into the bay of the ocean where these estuaries are. And they deplete the oxygen from this overloading of nutrients. And they cause a dead zone. They cause, you know, fish to die off and aquatic life to suffer. And so this was same thing in Japan. And so the thing about EM, you can brew this in really large quantities. You know, it's nothing to do this in, you know, we did a lot. I actually used 35-gallon barrels to brew EM, activated EM, super activated EM. And I'll tell you why. Some of you may find this interesting. We tried 55-gallon barrels, but I'll tell you what, you cannot move a 55-gallon barrel around unless you have a barrel dolly or a forklift. But a strong guy can move a 35-gallon barrel and pick it up and put it in the back of the pickup truck. So that's what I prefer using. But you can brew these in 250-gallon totes. You can do them in 1,000-gallon nursery, you know, containers or whatever. But they did this in Japan, and they started dumping massive quantities of EM into the creeks and streams behind industrial buildings. And they would go upstream, downstream. This is a key concept in probiotics, is upstream, downstream. And so through doing this, by remediating upstream all the way where the point source of pollution is, downstream, and working through the creeks and the pipes and the alleys, and it goes down and down and down and dumps into the ocean, they were actually able to renovate and repair the estuary and solve the dead zone problem and bring it back to life.
So there's another question in the chat. Is were you involved with the Lactobacillus experiments in aquaponics at KSU?
So Potent Ponics, I recognize your name, but the answer is no, I am not involved in that. I've heard of something they have going on over there, but I'm not involved in that, no. Although if you look online, you can see that I do have a publication on aquaponics when I worked with the ATRA Sustainable Ag program.
What about any more Vinnie Pinto stories?
Vinnie Pinto, he's kind of like a mad scientist. He did all kinds of these super activated EM concoctions. He's got a laboratory. I've been there to see it. he's just an interesting guy. He's definitely worth following.
What about, is there any way to find farmer communities or associations where these issues are actively pursued and practiced? That's from Jan.
Well, I think this is where, you know, I will say that the Korean natural farming is what's getting the real buzz right now. I did mention Chris Trump and Eric Weiner. These are two really top-notch teachers who are in Hawaii and Idaho, and they'll travel around, do workshops. And I think they're really doing a good job of teaching Korean natural farming. Communities, yeah, so Hawaii is really big. The Hilo area, the Big Island, is really big. There's just a lot of potential for more groups to adopt these things. I'd say we're still behind the curve, even though they've been around for decades.
All right, can you hear me? I'm unmuted, but of the three pathways, which do you prefer?
Well, I think that they're all okay. So let me mention another thing here. That's an option that farmers have, and I think it's important that we provide options, that we not try to pigeonhole people into doing it one way only. And so, composting is very common. It's widely known. It's widely practiced by organic farmers. It's a great tool for large scale, small scale, and it's just great. So, but the point is that you can take putrefactive situation or prevent a putrefactive situation by going either to aerobic composting or with fermentative anaerobic. It just depends on your situation. Now, I think one I didn't have time to include that, but I wanted to include a great meme. There's a great meme that it says, it's not the cow, it's the how. And what this meme is based on are these images of a fence line. On one side of the fence line is completely covered over with grasses and healthy forage. And the left side of the fence line is a barren wasteland. This land is all the same. This has been done in Africa. This has been done in the United States, in the Western United States. It's always the same. And the difference is the way that cattle were managed, livestock were managed. This is the decision that the land manager, the practitioner, the farmer makes. And so there's this notion that you should remove livestock from the land and leave it alone. And that is just incredibly short-sighted. What people like Alan Savory and the holistic grazers are saying is that put a bunch of cattle on there, move them through there like buffalo and let the land rest, and then it comes back around. This has been proven over and over again, holistic grazing. So it's not the cow, it's the how. This is the same exact thing that applies to sustainable agriculture, to nature farming, to organic farming, and to a conventional agriculture. It's like it's the same land, it's the same seed, it's the same water and sunlight. It's how you manage the system that leads to regenerative agriculture versus kind of the more rundown piece of land.
Where does Archaea come into all this? I almost never hear them mentioned, even they are major players, it is said, and that's from David M. Hall.
All right, Archaea. Now, this is interesting. There's a guy named, I think his name is Carl Oppenheimer. He was based in Texas, and he did a lot of work with Archaea bacteria in remediation of oil spills. And so he was really well known for this, and he had a terminology that I actually adopted and used to spread over into our work with large scale brewing of AEM and compost extracts and applying it to the land. And that the two terms that he came up with was bioaugmentation and biostimulation. This is really helpful to know. Bioaugmentation is going out with microbial food sources to apply to the land, or in his situation was oil spills. Say you got all this oil that has spilled onto the rocks, and it's the Alaskan Valdez, and it's, washing the West Coast, all the rocks are covered with all this black oil. What you would do is spray it with molasses and humic acid and seaweed extract and chitin, which are microbial foods, and they would stimulate the indigenous microorganisms to become active. And then they would gobble up the oil spill. By the same token, and you could do the same thing in agricultural land application. We did sprays of liquid compost extracts in a tank blend with all these good things, and we would spray it on pastures and orchards and vineyards and get great results. The other part of that is biostimulation. I'm sorry. Let me flip that around. Bioaugmentation includes the microbes, and biostimulation is where you add all the microbial food sources. So bioaugmentation is adding a boatload of microbes and spraying it out there. But anyways, that was Carl Oppenheimer's work. And yeah, archaea are just these primitive forms of microorganisms that have been around for billions of years. And they have some interesting properties. And yeah, there's one company that still makes them.
So one interesting thing about, you mentioned Robert Oppenheimer, he's mentioned in The Old Man and the Secret.
Oh, he is, yeah.
Yeah, so that's, I didn't realize he was the coiner of the terms bioaugmentation, biostimulation. That's awesome. And like you're saying, microbes can eat the oil spills, and there's hydrocarbons and carbohydrates. It's all, just need the enzymes and the acids and the microbes and the food for the microbes, right?
Well, we mentioned The Old Man and the Secret. We should mention that. That's James Martin in Texas. Texas has a history of these maverick people who worked with microbial systems. But James Martin, what he had, he would get a bucket or a tank, and he would put in seaweed. Ocean water, yeast. He'd put some manure in there and maybe some phototrophic bacteria in there, and he'd make this microbial concoction. And he would sell it to farmers. And so there's a number of companies in Texas that do that. There's still a company in Missouri that has James Martin's recipe I think it's called Microbial Solutions, LLC. And they've got James Martin recipe, and they sell those microbes.
Okay, so someone asked if you were, of the three major methods of harnessing biomass into fertility, if you were to introduce this to a place like Africa, what would you recommend?
Fermented plant extracts are so simple. Fermented plant extracts, look at what you can harvest from local native plants that have strong properties. And then also consider planting a special part of your garden into herbs that are grown for this purpose. It's really easy to grow turmeric and ginger, relatively easy. You can grow them in pots, you can grow them in the soil. I'm sure in Africa they would grow well. Those are strong herbal substances, garlic, peppers, a lot of herbs, comfrey, yarrow, a lot of native plants that have strong properties, trees with leaves and so forth. And all of those can be put into a barrel stuffed full of leafy biomass and then filled up with water. And then you would add either some mother culture microbes, or you would add some sugar to it. The biodynamic preparations serve the same purpose, and then do a fermentation. Some of these, depending on how you do it and what you're achieving, you're going to go more towards the stinky side, or you're going to go to the more sweet smelling side. It just kind of depends on how you manage that. I kind of prefer the sweet smelling side myself, but then you ferment those, and you're extracting all the good stuff out of the plants. You're generating all the microbial metabolites and bioactive substances, and then that material would be diluted, usually like 1 to 10 solution, for example, and used as a plant drench around plants for fertility and for tonics and for boosting plant health. You could also generate some of those are specially prepared for pest control. There's recipes on all that kind of stuff from both the EM experience and from the Korean natural farming and Jadam experience.
And then there was a question about if you knew of any research, which I would imagine would be like peer-reviewed sort of stuff about microbial synthesis of different biofertilizers, including Bokashi, IMO, Jadam, FPJ, all that.
Are there? Oh yeah, there's a growing number of science papers. A surprisingly good number of those are coming out of Indonesia on IMOs, and they're doing analysis of the IMOs, the efficacy of IMOs. I would say that, you know, because of the QSA Nature Farming international conferences and their proceedings, there's probably more literature on a single microbial inoculant related to EM than anything else in the whole body of agricultural literature. Yeah.
What about huge amounts of municipal compost here in South New Jersey, outside of Philadelphia, from street leaves and sewage waste? If you have 30,000 pounds, might you recommend any IMO improvements to municipal composting?
Well, it's really easy to manage compost if you understand all these processes. And, you know, that's like 15 tons. That's just not that much. That's just like what you can do on a farm or something like that. But anyways, yeah, there's just so many different ways to manage compost. And regular aerobic thermophilic methods work great. You can integrate EM with that. You can integrate IMOs with that. And people do all of the above.
Then what about just, this is a very complicated question, really, and very old question. When people, why, like when, and I guess why did, well, it says when people, I guess, when did people start to plow the soil and why? That's from someone named Axel.
Well, it's, you know, agriculture is very old. You know, one of the things I work with and teach is on how to produce really good transplants. And we like to use organic transplants because we have organic CSA on our farm. And I just think it's interesting to grow little seedlings, real quality seedlings, the right kind of potting mix, adding in vermicompost, all that kind of stuff. So I was looking that up one time, and it seems like you could look back and say that at least 6,000 years ago in China, they were raising seedling transplants of rice to transfer to the field. So this is something that pretty much every farmer knows about, especially if you're in horticulture, is raising seedlings. So I'm sorry, what was the front part of that question?
When did we start to plow the soil?
Oh yeah, so the same, by the same token, it's been around a long time. For 6,000 years, people have been plowing the soil, and they got a stick and a plow, and they got an oxen. Yeah, there's a lot of, I would say that I think no-till has a lot of uses, and it's really appealing. I think you have to match that with your farm. The smaller the scale you are, probably the easier it is to adopt no-till. The larger you get, it's probably not as practical. A lot of market farmers do some kind of tillage, but they're using the right kind of tillage. It's not improper. There's a thing called surface tillage, which is not no-till at all, but there's a huge part of the no-till market farming movement that do surface tillage. And it's just barely scratching the first top 4 inches of the soil, not turning it over and all that kind of stuff. So I think what's important, whether you're doing no-till or you're doing some kind of tillage, is to do proper tillage, understand what you're doing, and then add in a whole bunch of organic matter management. And the European concept around that is called humus management. And for example, you mentioned the Lübke family in Austria. That's another system that I've worked with. And I actually went over there, and that whole concept of humus management is really central. And that is that, yeah, you've got some tillage going because you're growing vegetables and you're transplanting into the soil. You're digging carrots and garlic and potatoes out of the soil. You're totally disturbing the soil in the process of doing that. But you're always using cover crops. You're using some compost. You're using some microbiology. You're always keeping the soil covered with living plants, some cover crops or vegetable plants or whatever crop you're growing.
Okay, because you have so much knowledge about biodynamics as well, have you ever seen the copper two-tiered plow that's supposed to mimic the mole movement?
Yeah, that's the Schauberger plow. Actually,
Yeah.
no, I have not seen one in practice. I don't have any in practice with that, but I've got, let's see. So Lala Gardens also wants to know what makes it super activated. Well, that's, again, that's the term that I use, I think kind of comes out of Vinnie Pinto, experience, but the EM, mother culture, and molasses is usually done in a fermentation at 1 to 1 to 20. So, you take 1,000 milliliters, for example, and that's the total amount of solution with water, 20 milliliters of mother culture, 20 milliliters of molasses. We would do like a 5-gallon barrel or, whatever, and we would ferment it with a heat pad at 95 degrees for 2 weeks. And we would extend and activate this whole amount into a rich solution of living microorganisms. So the super activated includes, you would now include EM ceramic powder and rock dust or ocean minerals, and something like some fruit juices and herbs. Alfalfa and yarrow are awesome herbs to use in an EM fermentation. I have some bottles in my garage that are like 12 years old that still have living microorganisms in them because these herbs were fermented in there. And so they're constantly regenerating. That's why they call them syntrophic microorganisms. They're constantly regenerating because of the substances that are in there. Yeah. So yeah, we're about to wrap up, and I'm not sure who your next speaker is.
So my next speaker on the agenda is Lance Hancherow from Saskatchewan.
Well, it's going to be coming up in 3 minutes.
Yeah. So maybe we're going to take a few-minute break, and I will switch him over as the host and get ready to bring on the next fellow. So thank you very much, Steve, for sharing with us everything. I have a bunch of questions still in my head, and I know there's other questions still, but it was very awesome to hear you today. It was an honor. Thank you.
Well, thank you, and I really am honored to be here with everyone else that's going to be speaking. And I know some of these people personally, and also through the network, through the internet. We've corresponded by email, but we've never shared an actual conference together. So I look forward to hearing the other speakers. And there you go. Thank you.